Okay, this is the second half of the bacteria of the skin. Now, if you recall, last time you took a red mannitol salt agar plate and you did the bacteria from your skin and you spread it on one half and then you pulled it through with a loop. Now, if it remains red, that means that it did not ferment mannitol. But if the media turns yellow around the colonies, that means that it ferments the mannitol. So this plate right here is actually not from your class, but it's an interesting plate. If you'll notice, half of this is one person and half is the other person, so whoever did this must have done two different people at one time or two different places. But see how this side is yellow? That yellow indicates fermentation. Do you remember our fermentation tubes? Red to yellow means acid production. So this is mannitol fermentation on this side. This is not on this side. So what you're going to do is, the first thing you're going to do is you're going to gram stain these. So go back to, I think it was page 93 or 35. Not even close. Page 35, 36, that's the procedure for the gram stain. The reason that you want to do the gram stain first is, if you look on page 173, it says, examine the colonies, record the appearance of mannitol fermentation, and then it says to gram stain it. Then it says after gram staining you do a catalase test. And if you recall, a catalase test means you add hydrogen peroxide and you look for the bubbles. So this time for doing the catalase test, you're actually going to do it on a slide. Yeah, so you take a toothpick. You're going to take a toothpick, and I'm not going to open this up and do that, but you're going to pick a colony up, put it on a slide, add your hydrogen peroxide, and look for bubbling. Okay, that's the catalase. Now you do both of those first, because if you look on page 174, it says subculture catalase positive, gram positive coccus onto another plate. So after you've done the gram stain and it's catalase positive, it bubbles, it's gram positive coccus under the microscope, then you're going to put it on a new plate and you're going to grow it for one more week. Okay? And that's because the reason to do this whole experiment is to isolate a catalase positive, gram positive bacteria. Now, the next thing that you're going to determine what you're going to do is you're going to look on pages 188 and 190. Or let's see, 188, yeah, and 190. These two appendices will give you kind of a dichotomous key to follow to determine what you need to do next. Okay? If you look on page 188, if you look on it on the left side, it says coccus. On the right, it says rods. So if you follow the coccus, that's what you had, and it's catalase positive, you go to the left. If it's catalase negative, you go to the right. Okay? Under the catalase positive, you have a choice of doing acid from glucose. So in order to determine if it has acid from glucose, you have to do a glucose fermentation tube. Once again, you'll just take the bacteria, put it in the tube, let it incubate till the next week, okay? And then if it had acid from mannitol or if it was negative for glucose, so you're not going to finish this lab for another week, okay? So this is like a 3-week or 4-week once you add our holiday. The other one down there at the bottom says acid from trihulose, which is also what the other flow chart on page 190 says. So if you look on number 1, it says cells arranged in tetrads. So when you look at the gram stain, look at the arrangement of cells. If they look like they're in 4s, that's a tetrad. The next one is a grape-like. Is it a staphylo? Okay. Glucose fermented. So the next time we come back, you see if this turned yellow. If it did, it's either Staph aureus or it's going to be one of these other Staphylococcus. Okay. That's why you're going to do the trihulose tube, because acid from xylose will turn this one, the trihulose tube, yellow, okay? And then no acid from mannitol, so if this did not turn yellow, then it's either Staph saprophyticus or epidermidis. And if you look back on page 188, acid from trihulose, so if this turns yellow, it's Staph saprophyticus. If it's negative, it's Staph epidermidis. So by the time that you're done with this next week, you will have the exact species name of the bacteria that you grew from your skin, okay? So the other thing that you want to look at if you need to do is the coagulase test. So if you look If you look on the one on page 190, after acid from mannitol, if it's coagulase positive, it's Staph aureus. If it's coagulase negative, it's one of those other ones. So if you go back to the procedure on page 174, it says to test for coagulase, you place a loop full of the coagulase, and it's going to be in a little tube. This isn't the tube. It'll probably be smaller than that. And it says to take a loop full of it. Take a couple of loopfuls, and you put it on the slide, and then you get your bacteria and you mix it in with there. And if it clumps into a heavy suspension, that's coagulase positive. If you think of like the blood typing, the reaction between cells when you mix the wrong blood type, you get that agglutination. That's what you're looking for that. So if it does that, turn yellow on mannitol, you have Staph aureus, and then all the other ones. I didn't see a lot of yellow in our class. Guys didn't come up with much Staph aureus, but that's okay. So once again, this is going to go for another week. We're just going to have more preliminary tests that we're going to do. The gram stain, the catalase stain, the gram stain, the catalase test, the coagulase test, and then do the 2 fermentation tubes. But you have to do them in the right order.